Talk more about structured water. You talked about the ATP piece and how that's not doing what most people are saying it's doing. Bring that in, and then bring the structured water piece in, and then we're going to tie this into the sunlight and then get into the grounding.
Yeah. Like I said, we all learn in school that we're harvesting this energy from the chemical bonds of food, and we extract electrons from that, and then we use those electrons to pass down this electron transport chain. And during that process, we get hydrogen ions that produced. They go up to the inner mitochondrial membrane space. Um, we get um, we get water produced. We get heat, infrared light. We get reactive oxygen species, and we also get ATP. We get all these different things produced as these electrons are passed down these proteins. And ATP holds it's a storage form of energy, so it holds this chemical energy in there. And so that storage form of energy, people think, oh, that must be the energy currency of the cell. It's what supplies all the energy for the cell. However, Gilbert Ling is a scientist that worked a lot with water. His work is actually what led to the development of MRI machines and how you can rearrange the water molecules and get a picture like it can using a magnetic field. And so yeah. But anyways, he also showed in his work that the amount of ATP that could be produced by mitochondria in a cell, any given cell, there's no way it could produce enough ATP to power everything that needs to be powered in the cell. He said that it couldn't even produce enough to power the sodium potassium pump, if that exists, which I'm not sure that it does. But he did mathematically and experimentally prove this, that there's no way you can make enough ATP to do that. So ATP, it turns out one thing that it does do is it does hold an energy bond. And that energy bond is used to unfold proteins in the cell. Because you get a protein, it's like if it's all folded up on itself, it's kind of like a tinfoil that's balled up into a ball, right? And so there's very little surface area when it's balled up like that. If you break it open and you unfold it, you get a much more surface area of tinfoil. So that's what proteins are like. They're all folded up, and the ATP comes in there and unfolds the proteins and makes them these big things with lots of surface area. Because what do you need to form structured water? You need a hydrophilic surface. And so there are studies that show that water in the cell is no more than zero point 0.5 nanometers away from any biological surface, or it should be if the cell is healthy. And so that's what ATP is doing. It's unfolding proteins. It's unfolding the cytoskeleton of the cell, or the microtubercular lattice, or whatever you want to call it, in the cell, creating surface area so that the metabolic water that's created by the mitochondria can go onto there and have a surface area to form structured water. Because the structured water is what holds the body's charge, because it's electronegatively charged. And Dr. Pollack has shown that many times in many studies that he's published, and many other authors aside from him have as well. And so that's what holds the voltage of the cell. We all know that, well, anybody who studied cellular physiology knows that the cell holds this voltage, this negative charge, so to speak, this pH of like 7.4 or something like that, slightly alkaline. It's like alkaline means more negative charge, not acidic or positive. And that's what does it. But we have to have this process happening. So ATP is still critically important, because without it, you don't get unfolded proteins. If you don't get unfolded proteins, you get less structured water, you get less cell voltage. And less cell voltage has been shown to be pathology. Cells that are diseased have lower cell voltage than they should. And that's the role of ATP, is to do that. It's a primary role.
In the three primary roles, ways I should say, of making ATP: the food, the sunlight, and the grounding. Any other big ones that contribute?
Those are the big ones that help produce the ATP. But again, like, the ATP is not the energy storage form. The energy storage form is structured water, and ATP plays a role in that.
No, that's what I'm getting at. To
Yeah,
open up the proteins.
yeah. So yeah, and all we're trying to do, like you said, is harvest that energy in the form of electrons from our environment. We can do that for way more than just food. So those things, like you mentioned, are the primary way we get electrons. But we also want to remove the things that interfere with them. interfere with the body's ability um, to harvest and to hold on to those electrons. So things that have been shown, so things that have directly been shown to break down structured water in experiments published by Dr. Joe Pollack are wireless signals. So specifically, a wireless router is what they used in his study, and it broke down structured water, I think, 10 to 15%, or interfered with the ability to form. And then the other one they tested was glyphosate, which is the herbicide Roundup. And so while we don't know for sure, there's probably other toxins and things that also do this. But if you think about it, since structured water is so negatively charged and it's holding on, like it's negatively charged because the oxygen is a bigger molecule and it's holding a negative charge, and there's not two hydrogens to balance it out anymore. There's only one. So that's what gives it the negative charge. But it's also negatively charged because it's a storage form of electrons. Because the water molecules are now kind of linked together, kind of like an ice, but without the extra hydrogen, electrons can travel through them, right? So electrons are traveling through it, and that's what gives it this negative charge. That's why it's a storage form of it. And so if you interfere with that, you bring down the body's negative charge. But since it has this negative charge, we all have heard that oxidative stress or inflammation or things like that, those things steal electrons from the body. And the place that it would steal electrons would be structured water, because that's where the electrons are stored. And so that makes sense that when you look at what glyphosate is and how it acts like it's one thing that can cause inflammation, oxidative stress, this kind of stuff. It's just stealing that charge. So anything we know that acts like oxidative stress, like heavy metals in the body, could act like that. Or when we get poor metabolic health and we're producing too many reactive oxygen species, like all that stuff could contribute to this destruction of structured water and then low voltage in a cell. And that's just well-known science. The oxidative stress breaks down or steals electrons from tissues, and it steals it from structured water. So then, wireless router and toxins. So we want to remove the things that steal the charge. We need the raw material, which is water, which we make from mitochondria or we're drinking for our bloodstream and lymphatic system and stuff. We need to expose ourselves to the things that build structured water, infrared light mainly, but sunlight is the original form, and grounding, and then healthy metabolism. So we're extracting electrons from that, and then we remove the things that break it down. And if you look at that recipe, it's like, and look at the modern way of life, it's a bad recipe, right? That's exactly what we're doing, where we create poor metabolic health. We're not drinking enough water. We're drinking these dehydrating things, a bunch of caffeine and sodas and things like that. We're not living outside with feet contact with the earth in sunlight, and we're surrounded by artificial blue light and EMFs and exposed to so many toxins. And it's just like, no wonder our bodies are struggling. That's and so when you talk about pain, like that's going to significantly increase the likelihood that cell voltage drops. And what happens when cell voltage drops? It's one thing that your body says, hey, what's wrong with this tissue? I'm going to send a pain signal there. So that threshold of what's going to cause pain is drastically increased.
Let's talk about the next step of this story. So you have your mitochondria making ATP, unfolding proteins, structured water is forming inside the cell. How does that energy then get used?
The cellular energy, like the structured water energy?
We used to think of it as ATP, but now that we're saying the energy is actually the structured water, how is that used by the body? The next step.
Yeah, so this goes down to like phase transitions. Well, first of all, structured water is a battery. So it's when structured water forms, there's an area of negative charge, and then because of the way it forms, it creates an area of positive charge next to it. And that positive next to a negative creates an energy gradient that it stores energy. It basically, just like a battery, stores energy. There's a positive and negative into a battery. We're concentrating energy in that area. And so this is what structured water does. And so when you form structured water on this protein in the cell, you've created a battery right there. And in Dr. Pollack's lab, they've shown that you can plug, like they make a structured water zone and they put an electrode into the structured water and into the positive area next to it in the bulk water, and you can power a light bulb. So you're creating energy, you're creating electricity. And so it's stored right there. So when you think about this, it's not like all the cellular proteins are unfolded and everywhere in the cell is full of structured water. The majority of it is, but not all of it. And so there's still these little pockets of things where there's not structured water, or there's still these little pathways. And we talked about in our last episode how this does the work of moving fluid. This energy gradient does the work of moving fluid, and it's the same thing in the cell. That's what kind of keeps the fluid moving through cellular pathways. And this happens even in embryos where there's no heart function yet. That's how fluid starts moving initially in embryos. So that's what's creating this fluid movement. And then we also get what are called phase transitions. And so Dr. Pollack wrote a very good book, very well cited book called Cells, Gels, and the Engines of Life. And this book basically makes an argument that physiology happens through phase transitions. What I mean by phase transition is, like, we talk about structured water, it's actually the fourth phase of water. And so when we talk about a phase transition, it's going from a liquid phase to a structured phase and vice versa. And when that happens, cellular physiology happens, so we start to see action potential. So we learned in school that an action potential is when this depolarization of a cell happens. So depolarization means it loses its charge temporarily. How does it lose its charge? A phase transition. It loses structured water temporarily, and then it comes back, right? And that phase transition, there's many things that can signal a phase transition. It could be a change in pH. It could be a temperature change. It could be anything, all these different things. And it signals for a phase transition. And that tells the cell to do physiology. And the cell that happens here, then it happens to the cell next to it. And it's like this, it just happens super fast. And that's what this depolarization is. And then repolarization of a cell is. That's what we see. So that's how physiology happens. And that's how the body uses that energy to hold the charge and then create phase changes in response to external stimuli.
Got it. That was what I was getting at. So phase transition is when this structured water is actually used to do something in the body. And that's when it goes from being structured water to a liquid again, and then it goes back.
Mm-hmm. Yeah.
And all that happens, I'm assuming, super quick.
Yeah. Like faster than you could probably think. Like it's so fast. But that's what, like, everybody who studies physiology in school, we study like nerve conduction and action potentials. That's what we're studying. We're saying, oh, this depolarization happens in the cell and then it repolarizes and it happens really quick. And that's what sends a signal down like a nerve or whatever, this action potential. When in reality, it's just, it's like a continuous phase transition that's happening, and it's happening instantaneously. Like probably happened 1,000 times in the last sentence I just said. It's how physiology happens. And like, again, Dr. Pollack in his book, he showed how phase transitions are responsible for muscle contraction, nerve signals, and various other things, parts of physiology. That's just the evidence we have so far.
If you enjoyed that clip, you're going to want to head over here and catch the full episode. I'll see you over there.
How much serotonin you make when you get UVA light in mid-morning is going to dictate how much melatonin you can make later when you block blue light after sunset. So the storage of serotonin, that's going to be converted to melatonin later. So I tell people